Blood typing involves identifying specific glycoproteins, called antigens, on the plasma membrane of the erythrocyte. The antigens give us the ABO blood grouping system in addition to the Rh factor. Individuals with A-type blood have the A antigen on the erythrocyte plasma membrane, while individuals with B-type blood have the B antigen. Individuals that have both the A and B antigens have the AB blood type, while individuals who have antigens other than A or B antigens have type O blood. It is possible to have both A and B antigens because humans have 2 copies of the blood type gene. So if one of those genes gives our blood the A antigen and the other gene gives our erythrocytes the B antigen, then that individual will have both the A and B antigens present on the erythrocyte plasma membrane. The other antigen is the Rh antigen, or the Rh factor. If the Rh antigen is present on the erythrocyte, then the individual is Rh positive, and if the Rh antigen is absent, then it is said that the individual is Rh negative. Figure 18.17 graphically shows the ABO antigens on the plasma membrane of the erythrocyte. So those individuals who are A-type blood have the A antigen present, while those who are B-type blood have the B antigen present. If both A and B antigens are present, then the individual is type AB blood. And if antigens other than A or B are present, then the individual has type O blood. To determine one's blood type, a small sample of blood is mixed with a fluid called the antiserum that contains antibodies against specific blood types. The A antiserum contains antibodies against A-type antigens. The B antiserum contains antibodies against the B-type antigens, and the D antiserum, which is also called the anti-Rh antiserum, contains antibodies against the Rh antigen or the Rh factor. The antiserum causes the erythrocytes of that specific blood type to clump, which is also called agglutinate. So for the purpose of blood transfusions, AB positive is what is known as the universal recipient, meaning that they can receive any blood type during a transfusion, while O negative is the universal donor, meaning that individuals who are O negative can donate their blood to any other blood type during a transfusion. So examining a random sample of 100 people, we see that having the Rh antigen is far more common than lacking the Rh antigen, and types O and A are some of the most common types of blood. Meanwhile, AB negative is the rarest blood type. Table 18.2 outlines the percentages of blood types among different populations of peoples in the United States. We see that O positive is the most common type of blood across all groups, with A positive not far behind. AB negative is the most rare, comprising far less than 1% of peoples in all groups. In the lab, we can determine blood type using the antiserums described earlier. In this example, we have placed 3 drops of blood from the same person within 3 separate circles on a blood typing card. To the drop of blood on the left, we have added a drop of anti-A antiserum. To the drop of blood in the middle, we have added a drop of anti-B antiserum. And to the drop of blood on the right, we have added the anti-D, which is also called the anti-Rh antiserum. We would then mix the blood with the drop of antiserum and wait several minutes. If the corresponding antigen is present on the red blood cells, they will clump or agglutinate in the presence of that antiserum. So in this example, the blood sample has agglutinated in the presence of the anti-A antiserum, which means those erythrocytes have the A antigen. They have not agglutinated in the presence of the anti-B antiserum, which means that they do not have the B antigen on their surface. And finally, we see that the sample of blood has agglutinated in the presence of the anti-D or anti-Rh antiserum, meaning that the erythrocytes have the Rh antigen present on the surface of their plasma membrane. Since these 3 drops of blood are from the same person, we would deduce that they have the A antigen and the Rh antigen present on their erythrocytes. So that means that they are the A positive blood type.